Thank you all for attending, and you guys, Marsha and the Human Society, for accepting me to talk on behalf of Daniel. So I'm going to talk a little bit about the work we have been doing in the lab of neuroproteomics, which is held in Unicamp. And I'm talking today a little bit about our past work and how we reach to working with human organoids right now. So mostly we work with neuropsychiatric disorders that Georgia already introduced a little bit. So it's nice to see this all complementing each other. And neuropsychiatric disorders are a really burden to all society. So most of the diseases, like here we see 28% are people that are affected by this disease in some part of their lifetime. And right now we have a lot on depression, bipolar. Several of the people working do have schizophrenia, and 25% of those people will have this over their time, even if they do have for us just some time or for their whole life. And this may come to shorten their lifespan by 10 years, because they do have problems and will maybe not survive as much as people would generally. And normally this mortality rate is increased in population with psychiatric disorders. Here I'm going to talk a little bit more about schizophrenia and the work we have done with schizophrenia, which has a focus on understanding the biochemical components which are involved in this disorder, and how we can lead some of the research and towards better treatment, because right now we do have some treatments which are not completely curing or even making the people working and being all the time focused. So about 40% of the patients do not have their work-life balance happening, although they do have treatment, which are the antipsychotics. And this is also something that we address here, which is diagnostic. So most is done only with clinical interviews, and we do not have any biomarker that can detect a disorder or differentiate one disorder from the other. For example, a bipolar, schizophrenia, or even depression, they do have similar traits, and then the prognosis. And we are looking then for translational strategies that we can overcome all these complex problems that we have here. Those disorders such as schizophrenia, depression, they do have several factors that are implying in them happening. So, for example, we are focusing right now on some of the neurodevelopmental factors, but there are several others which are environmental factors which can affect those people over their lifetime. And we are looking particularly to the proteome of brains and cells, which are a little bit behind, I would say, in the transcriptomics, comparing with talking about throughput. So we do observe in proteomics much less proteins comparing to genes that we can observe expressed. Right now we can look to about 20,000 genes that we have in our genome, but we are reaching something like 10,000 proteins that we can already observe while looking to a whole cell proteome. This, I mean, just looking to the whole lysate of a brain or a cell, we can have access to around 10,000 proteins. If we separate them in even smaller pieces, for example, a synaptosome, for example, if we just look to that small area of synapses, we can observe around 3,000 proteins being expressed and differentially regulated according to some diseases. So here we are looking for those quantitative proteomics, and so look into what are the differences that we can observe and how can we target those issues. So how we can see if a medication is acting right, and how can we do personalized medicine comparing to that. Like, how can we stratify patients and know which kind of antipsychotics, for example, is going to work in this type of patient. And of course, looking to disease mechanisms to look for new drugs that can act, and biomarker candidates. And here, for example, you can look to blood markers or even CSF markers that we can see how all those changes are being marked or shown. And we can look into quantitative proteomics or general shotgun, target multiplex proteomics, or just looking to some special proteins that we already know, and these are really important for biomarkers. And look at protein-protein interaction, because this can be different depending on the disease that you're looking for. And the modificomics, which is more or less how we are looking to cell-cell signaling and how one cell interact with the other. Some of the work that we have done, we are really trying to find some of those different tools and to help in psychiatric disorders, and like biomarkers and phosphoproteomics, exosomes, and stem cells. Because we want to understand a lot of those mechanisms, and these are just work from brain that we started with post-mortem brains and blood from different patients. So we can try to stratify all those different outcomes here. Working with different areas of the brain and taking a look on the whole proteome. So doing the proteome of each specific part of the brain, comparing diseased and controlled patients which do not have any other neuropsychiatric disorder. We could see a broad difference in the proteome of all those patients. These were mostly related to the metabolic pathway, mostly here in glycolysis. So we see the energy metabolism really being affected, and in schizophrenia, a lot is involved with glycolysis, which is really an early step pathway that we do have in our energy metabolism pathway. And here, from here, you already have some ROS production, and most of all this being affected and disrupted due to differential expressed proteins that are impaired here. Of course, we do have some other proteins which are related to oxidative phosphorylation, but glycolysis is still higher in schizophrenia. And also we can see that patients do have increased levels of pyruvate in their blood compared to controls. And this is because of the disrupted glycolysis cycle. Others have also shown this area of glucose tolerance and this involvement with insulin tolerance, and all this related to schizophrenia. But our question is always like, energy metabolism is always impaired in some diseases, and how specific could this be? And we want to know how this can be related. So, for example, here we in a recent review that we have done, we show that we are only looking to proteins which are related to energy metabolisms. And we are comparing schizophrenia, bipolar disorder, and major depression disorder, and show here that only 5 of those proteins are really expressed in all of these disorders, differentially expressed here. I'm not even looking to up or down regulation of them, but we do have major area here showing that depression is a lot related to the oxidative phosphorylation comparing to schizophrenia. So what we can learn from that is that we can look to different proteins which are related to those ones while talking about depression, while if you have schizophrenia, you should look to a upper pathway, which is glycolysis, and see if those differences could be found. And you can try to differentiate those cells according to that. So coming back to the global expression of those proteins, we want to then look to differences in cells which are related to those brains, which were all those works were done with post-mortem brains. So we want to translate them to a little bit to cell cultures. At first, we started doing this with cell lines. So we did have cell lines for neurons, oligodendrocytes, and astrocytes, and we want to see differences that we could find in all of those ones. So mostly because several of those proteins that are found also differentially expressed are related to oligodendrocytes. So from the whole brain, we found that several proteins which were altered together with all those energy metabolisms. Another one, other pathway or other biological process that was included was proteins which are related to myelin. So we wanted to see which are those differences. So here just showing these differences. And wanted to compare all those cells. We used a mechanism which was first described in mice. So we wanted to apply to cells, which is using an NMDA antagonist, MK801, which in animals gives a behavior of schizophrenia, how this can be related. Though as inhibitor of NMDA, we do have several outcomes here, and we wanted to check these on cells. So we did different cell cultures. Here we are showing a work done by Paul Guest. So we treated differently each cell and compared them. Like here we have neurons, or we have astrocytes, or oligodendrocytes. So the whole treatment was done in 3 different cell lines, which was having the control, not treating with anything, or treating with MK801 for 8 hours, and also with clozapine. Clozapine is antipsychotic, which is one of the most used and somehow one of the most effective, although has severe what's called the disease? I'm sorry, I forgot. Then we try to mimic a little bit with using clozapine, a post-mortem brain, because we never know what, using post-mortem brains, we do have the disease, but we do have the disease after the person is already dead, so it's never a tissue that is alive, and was treated with several antipsychotics that we sometimes know, and sometimes we don't even know which are those antipsychotics. So here we try to mimic this part. And we observed that neurons, we looked to specific proteins in this area, which are only shown differences when we are comparing them to clozapine. So mostly the effects on neurons that we were observing here on energy metabolism was related to the effect of the antipsychotic and not to the disease which was mined by the MK801. While oligodendrocytes, on the other hand, showed several of those proteins altered comparing with the disease or with the NMDA antagonism. And so we thought that oligodendrocytes should be a cell that would be interesting to look further. And maybe instead of only astrocytes, those oligodendrocytes were the ones that were altered in schizophrenia and not supplying the correct amount of energy to neurons, and not the neurons themselves that were being affected by this energy metabolism, but the glia which was surrounding it and providing their energy. And then we started to work with Stevens here in IDOR to make those cells, instead of working with mini brains and looking for this comparison of schizophrenia patients, and looking to astrocytes, neurons, and oligodendrocytes. So first, talking about we generated those organoids in spinner flags. So during 45 days, so we were working with really young and already developed tissue with around 45 days, and we did proteomic approaches from those. So all the time I'm going to show you the whole proteome of the organoids here. First, we did just checking which our different spinners were producing kind of the same types of cells. So according to protein, about 95% of the IDs of proteins that I'm having here, so about 4,000 good to analyze proteins, are the same among the whole bunch of organoids that I'm picking from different spinners or different cultures. And only that meaning that only 1% of proteins are different between all approaches that we can have here. And first, I'm going to show you something that Stevens already started yesterday, just to show you here, which was the derivation of astrocytes from those human organoids. So we had the whole 45 days organoids here, which were dissociated because they already had some glia cells being expressed. So we dissociated them and cultivate to eliminate all the other proteins. This was an approach to have one single cell type analyzed, one single cell type meaning astrocytes and the whole astrocyte types there. And we did a comparison of those by functionality and proteomics. I'm going to show you here the proteomics. So, as shown before, so we have several proteins that are really common among differential cell types. So here we have two different embryonic cell types, which we did the experiments, and we compared two human adult astrocytes. This was obtained by surgery, so we did the same extraction, so dissociation of a small piece of brain, which was removed from surgery, and which are really rare to have. And we could compare these two derived here, comparing to these human astrocytes. And although we do have several common ones, here in this type of correlation, we can show that not only the types of proteins are similar to those astrocytes, or about 0.7, the 70%, I would say. It's also similar in comparison with intensity of the protein that is expressed. So the differences relies on mostly on developmental proteins that the human astrocytes do not have than in other ones that we cannot directly compare. And we do have the same amount of GFAP, vimentin, LDH, which are proteins that are related to glia and astrocytes. And we observe them in both cells, the ones that we derived and the ones that were from surgery. Several pathways also are really similar. This, I mean that proteins that were belonging to integrin signaling or important glycolysis, they were found in both specimens. So this was reproducible and was important to show that we can work with those astrocytes that are iPS-derived or embryonic stem cell derived to mine an astrocyte or to have astrocytes to work with. And also looking to the whole organoid, to the proteome of the whole organoid, we can observe other proteins. So here we already have, as I said, GFAP, vimentin, and others, which are related to astrocytes, and from that we derived more astrocytes here. And we observe a lot more proteins, and here I even have smaller comparing to the other ones just to fit my figure. But we do have in those days, like 45 days, we do have much more proteins related to neurons and several types of neurons that we can observe here. But I would say that we already observed some oligodendrocytes proteins related, which are not only interesting here, for example, CNP, which is from progenitors, but also several which are myelin related to showing that those organoids, they are starting to produce proteins which are related to those cells. And also a little bit of microglia-related proteins like coronine C and so on. So we showed later on that those cells, when we dissociate them, we can see several other proteins being expressed in those cells, such as IBA1, CD45, which are then coming from those organoids. And we do have proteins which are related to synapses and all those clustering, and we can work with this model as well, and protein-based level, that we can show proteins those organoids are doing synapses already. And here I'm showing just those are rare to find, but those we do observe some myelin starting to be formed in organoids. So those are 60-day organoids, which are showing here the myelin, which are backed up by the proteins that we already found before, as I showed you, all the myelin proteolipid proteins that are there. And I would like to thank you all and thank the lab of neuroproteomics, which is there, all the collaborators here in the DOR Institute. And of course, you can find us on Facebook and have questions. We do keep a page that we try to bring all those news and bring people together with science and to discuss all that, our funders. And please come to visit us at Unicamp in Campinas. And thank you very much.